SNMMI 2022 annual meeting is recognized as the premier educational, scientific, research, and networking event in nuclear medicine and molecular imaging. The four-day event, taking place each June, provides physicians, technologists, pharmacists, laboratory professionals, and scientists with an in-depth view of the latest research and development in the field, as well as providing insights into practical applications for the clinic. SNMMI TV starts right now. Hello and welcome to SNMMI TV. My name is Gina Phillips and I'm here to share with you all the latest at this year's annual meeting. Stay tuned as we'll be having one-on-one conversations with leaders in the field. We'll be highlighting organizations that are at the forefront of nuclear medicine and molecular imaging. But first, let's take a deep dive into therapy and diagnosis for thoracic malignancies in the breast and the lungs.
In these sessions, they're going to highlight some of the novel PET probes or molecular imaging probes for thoracic malignancies, including breast cancers, and how they fit in the overall management of these malignancies. And that's really a key take of these sessions. Another thing is we're also introducing or discussing something called hybrid imaging, which is a marriage of two imaging modalities, one functional, one anatomic, and for example PET-MR, and that is really another key point of this session. I'm most excited about sessions or topics that will be discussed that show response assessment, which I think is a very crucial need of our times. Most malignancies, including thoracic and breast malignancies, now have a vast array of novel therapy agents, and all these therapeutic agents are quite expensive, and not all patients respond to these. So anything that's going to help us assess or find consistently and accurately responding patients from non-responding patients, that's going to help us a lot in this current environment of fiscal conservatism for healthcare, as well as it prevents toxicity and unnecessary treatment. So I'm really excited about that. From this session, I think attendees should take away the message that we are just embarking on this exciting journey of molecular imaging. It started with FDG PET, and FDG was aptly named the molecule of the century by some, and we are still eking out the nuances of FDG. But with these new molecular imaging probes that are very specific for certain aspects of cancer, we could finally move closer towards our goal of P4 medicine. I also hope that the attendees take away the fact that with technological advances, it will be easier and feasible for us to get high quality scans with significantly lower radiation dose, and that's another very crucial aspect to our field.
Radiopharmaceutical therapy has grown rapidly in the past several years, and so we want to hear from members about how their institutions or practices have responded to this rapid growth. Let's take a look.
So we're really excited about the growth in radiopharmaceuticals over the last decade. We are really now at a tipping point where we can not only help patients by detecting disease earlier, but treating them now with radiopharmaceuticals. So as a group that's developing novel radiopharmaceuticals, we've now been focusing our efforts on the unmet need in diseases such as pancreatic cancer.
So one of the ways that we've adapted to the growth in radiopharmaceuticals at Sloan Kettering is by expanding our capability to make alpha GMP radiopharmaceuticals.
For us it's very important because we are developing a medical imaging system that images those radiopharmaceuticals. So the fact that they target, those radiopharmaceuticals target much better tumors, lesions, problems in the brain, function in the brain, function of the lungs, that allow us to image with a very high image quality those agents. And on their side, hopefully, so we're in startup, so we are just developing, it's not FDA approved yet, but that our system will allow the radiopharmaceutical developers to actually image and see where those radio agents are concentrating and going with a sub-millimeter resolution.
Definitely embraced this a new therapeutic approach which is actually not that very new because nuclear medicine physician starts with therapy with iodine, one of the historical cases. But now we're having the amazing opportunity to use the same nuclides, the same radiopharmaceuticals with nuclides for diagnostic and then label them with nuclides for therapy. And so we have this theranostic approach which allows us to see and target directly the disease. That's for instance for cancer in prostate or neuroendocrine tumor and so forth. So when you have a specific target to the tumor cells, you can actually label it with specific nuclides which use their bullets, let's say, to kill the cancer cells.
I am joined now by Andrew Scott. Thanks for sitting down with me this morning.
Thank you.
How are you doing so far?
I'm good, thank you.
Great. You've taken in some good sessions, chatted with some colleagues.
Yes,
Yeah.
I mean, it's early in the meeting, but the opportunity to actually meet people for the first time in 2 years in person has been terrific.
I know.
And, looking forward to the rest of the meeting.
Excellent. I want to start off talking about some challenges in oncology and therapy right now.
Well, I think that aligned with opportunities are always challenges. And so I think the key issues that we need to face at the moment are how do we develop the evidence to be able to, for nuclear medicine imaging and therapy, obtain the approvals and the funding that we need for the opportunities that we can see that are there. Right. We know, for example, that there are imaging studies which are approved in the US, which aren't approved in other countries, and vice versa. So I think being able to generate the data and to be able to use that to achieve the regulatory and funding approvals are one of the main challenges. One of the programs that I'm involved with at the moment is with the International Atomic Energy Agency, particularly looking at access and availability for imaging and therapy for nuclear medicine in low and middle income countries, which, when you think about it, is 3/4 of the world's population. And so we're looking at ways we can try and improve that, and we have a number of initiatives at this stage we're pursuing.
What are some opportunities that you mentioned that you're looking
Yes.
forward to making happen, or you're
Yes.
seeing things open up right now?
Well, I think that we're in probably one of the most positive stages of nuclear medicine that I've seen in my career. The number of new imaging and therapeutic opportunities are enormous. The dotatate and the PSMA therapies that have been approved in the US, poised for PSMA in Europe and for the rest of the world waiting to be able to get access on a more routine basis, is really going to transform the way that nuclear medicine is practiced. And I think from an opportunity perspective, that's enormous. But we have some challenges there too. So how do we have the workforce that we need? How do we get access to some of these new therapeutics in ways that will provide equity for patients that really need the treatments? From an imaging perspective, again, I think we're poised for enormous progress as well. I mean, PET clearly continues to expand. There's so many new traces which are being investigated in oncology as well as in many other types of nuclear medicine indications. And I think that those also will continue to expand into the future as well.
Wow, very exciting.
Yeah.
Anything that you haven't mentioned that you're looking forward to learning more about or you're particularly excited about?
Well, I think that there are some really exciting innovations in both technology as well as in traces, which will be presented at this meeting that I'm very much looking forward to. So from a technology perspective, I think some of the newer generation digital PET and SPECT cameras are very exciting. Certainly the whole body systems are looking incredibly exciting, although they're quite expensive, so not everyone will be able to access them. But also from the chemistry side, looking at the ways that new synthesis techniques can be used and analysis with AI machine learning. I think there's some huge advances being developed there as well.
Well, fascinating. I hope you have a great next few days.
Okay, thank
Thanks
you.
for sitting down with me.
Thank you very much.
The Sierra trial has set out to transform the paradigm of bone marrow transplants with the use of targeted radiotherapy for conditioning. This has the potential to provide the option of transplant to patients who may not qualify under the current standard of care. Inium Pharmaceuticals thinks there may also be a future use in other hematologic malignancies, solid tumors, and as a part of the regimen for cell and gene therapy. Let's take a closer look.
The Sierra trial is a study of a novel agent that incorporates targeted radiation as part of the conditioning regimen for a stem cell or bone marrow transplant. The IOMAB treatment is being used to treat patients who are older with refractory leukemia, for which transplant ordinarily is not an option. Pound for pound, a easier treatment to deliver than our regular strong transplant therapies. The rates of sepsis and mucositis seem to be lower. It's worked very well to get patients into remission. So it's a very exciting treatment.
It is definitely groundbreaking because this is one of the few treatments where we have been able to integrate dosimetry by doing patient-specific calculations and giving these radiation doses within the prescribed limits. I think it has the potential for application in other areas. It can be used for therapies such as the cellular therapies and gene therapies.
If it proves to be a superior therapy to conventional care, the transplant community will start using the agent more frequently.
Inium Pharmaceuticals, Inc. is a clinical stage biopharmaceutical company developing targeted radiotherapies. It combines the targeting ability of molecules, like antibodies, with the cell-killing ability of radiation. Actinium Pharmaceuticals is also committed to staying at the forefront of innovation, collaborating with leading global biopharmaceuticals like Astellas, Inc., using its targeted radiotherapies as part of the regimen for cell and gene therapies.
Actinium was formed to harness the power of radiation, making the lives of cancer patients better.
In the Sierra trial, we're able to take patients who have been heavily pretreated and we're able to successfully bring them to transplant using IMAB as conditioning prior to the transplant.
If it proves to be a superior therapy to conventional care, the transplant community will start using the agent more frequently. Pound for pound, a easier treatment to deliver than our regular strong transplant therapies. The rates of sepsis and mucositis seem to be lower.
We shipped out hundreds of radioactive drugs without missing one single dose.
It is definitely groundbreaking because this is one of the few treatments where we have been able to integrate dosimetry.
Inium Pharmaceuticals have an active and ongoing collaboration with Astellas. We also look at opportunities to take our radioconjugates into solid tumor space.
One of the other studies that we have underway are efforts to utilize IMAB as conditioning prior to CAR T-cell therapies.
We really look forward to changing the paradigm of how radioisotopes are used, where we believe we can take a leadership role.
I'm sitting down now with Julie Price. Thank you so much for chatting with me today.
Glad to be here.
Let's talk about neuroscience. What are some challenges that you're currently facing?
Well, I would say it's a complex field. We have new technology, new applications in neurodegeneration is an area that we focus on. I think training the next generation is our duty, but also our challenge to engage them in our research to help them build the future in neurosciences, in nuclear medicine.
Absolutely. Are there opportunities that are popping up that maybe are unexpected?
Well, there are opportunities in basic training grants, fellowships. Certainly where I am now, we're working hard to build further opportunity from an institutional level, but also many of our research opportunities are funded by the National Institutes of Health Foundations and liaisons with pharmaceutical companies.
Okay.
So there are opportunities.
And as far as this year's meeting goes, what are some highlights?
I would say certainly there is still lots of interest and productivity in imaging of proteinopathies, tau and amyloid, neurodegeneration. They're very interesting applications in psychiatry. It's a smaller section of our overall abstracts. But we've seen a very interesting emergence of total body PET as well. There's applications in neurodegeneration, and that will be presented at one of our integrated sessions, integrated session 8 on Monday. Also, there's the Pen Explorer total body scanner. They have applications involving muscarinic receptor and nicotinic acetylcholine receptor in smoking. So I think the application of the newer technologies is quite interesting, in addition to the work that's going on in neurodegeneration as well.
Has it been nice to be here in Vancouver in person with your colleagues?
Yes.
I imagine it's a relief to get off the screen for a bit.
Most definitely.
Well, thanks for sitting down with me today.
Thank you.
All the
Nice
best.
to meet you.
Joining me now is Simon Cherry. Simon, thank you so much for sitting down with me today.
It's a pleasure.
How is it all going so far?
It's wonderful to be here, back in person again after a couple of years, and beautiful location as well.
Isn't it? I
Yes.
know. Oh, and we're right by the water right now.
Yes.
It's lovely. So your plenary is coming up.
It is.
Very exciting. I want you to give me a little sneak peek about it. So you're exploring time and its role in nuclear medicine. What is that all about?
So I chose the theme of time for a couple of reasons. So first of all, I want to pay tribute to some of the people that laid the foundations for our field. So I want to look back in time and talk about some of the seminal work that led to where we are today. But also time is such a fundamental part of nuclear medicine. When we inject our radio tracers into a patient, the distribution is constantly changing with time. The image you get from 1 minute to the next is different. And so I want to explore how we can use that information and to gain better insight into the underlying biology. And then lastly, I want to go to the real extremes of time and look at detectors that can measure time with a precision of less than a billionth of a second, which is what we use in time of flight PET imaging. And we're now moving towards a situation where we can actually directly generate images using the timing information. And I think that's going to be the future in terms of the technology. So I want to explore that a little bit as well in the presentation.
That's amazing. Oh my gosh. What have been some of the historical developments?
So I think, obviously you start with Marie Curie, probably, isolating different radionuclides, really setting the stage of being able to use radionuclides in practical applications. Following from that, George Hevesy, in the 1920s, using one of those radionuclides, lead-212, to study its uptake in plants. That was the first application of a radionuclide to a biological system. He then extended that work into animals, and so that's really the basis of the tracer kinetic principle, where you put in trace amounts for radioactive material and watch what happens in a living organism, first in his case plants and then into animals, and of course now we do this in humans. And then finally the people that started to develop the technology, the imaging technology, so Benedict Casson, whom the lecture that I'm giving tomorrow is named after. He really developed the first directional imaging detector and then scanned that across the thyroid to produce the first image inside of the human body. And then Hal Anger, who took that even further to develop the first gamma camera, also called the Anger camera in his name. And of course, that was the start of imaging technology, which is what my career has been based on. Wow. So those are really the people that I look to for inspiration.
Wow. Your lecture is going to be fascinating, I have a feeling. Thank you for that sneak peek. What does nuclear imaging look like right now, if you had to sum it up?
Well, we're at a very exciting point, as we've seen already in some of the plenary sessions, this revolution in theranostics, so having diagnostic and therapeutic capabilities, massive growth in the field in that area, very, very exciting. On the technology side, we have incredibly sophisticated technology now, and I really want to recognize a lot of the scientists and engineers in industry who are often the unsung heroes who develop these incredible machines that are robust and reliable and safe that we use in our hospitals every day. But there's still more we can do. And so I think although our imaging machines are fantastic, we can still make them better. So we can see disease earlier, we can quantify disease better. And of course, we'd like to bring the cost down as well.
When you talk about the future of nuclear imaging, what does that hold as far as a global picture?
Yeah, I think that's a really good question. I think we need to recognize that the needs for nuclear medicine are going to be different in different parts of the world. So a lot of the focus here has been on oncology, for example, but in other parts of the world, infectious disease is probably a much bigger issue. So I think expanding the reach of nuclear medicine to other disease areas that are broadly applicable across the world. And then in resource-limited areas, it's a problem how we get nuclear medicine accessible. It's not the easiest technology to disseminate, so I think we need some clever ideas about how we can make it much more globally accessible.
Simon Cherry, thank you so much for breaking it all down for us.
You're very welcome. Thank you.
Good luck with the plenary.
Thank you. Thank you very much.
And now for a patient's perspective. We're joined virtually by Van LaCour. Van, thank you so much for sitting down with me today. You've had quite a journey with cancer, so take me through it. When were you first diagnosed with prostate cancer, and what did treatment look like for you early on?
I was diagnosed with prostate cancer back in November of 2007. I had gone to my urologist for a checkup. And when he checked, he found that my prostate was swollen, and which gave him great concern. And he wanted to do a biopsy, did a biopsy. Sure enough, it came back positive for cancer. Biggest problem, it was swollen. It was close to the outer wall of the prostate. And his concern was it could erupt and go to other areas of the body. So the decision was made to do the prostatectomy and do the surgery and remove it, and lessen the chances of the cancer spreading and what have you. Well, after the surgery, he determined that I was cancer-free. And 8 years later, the cancer reappeared. And we went through series of doing the chemo and some oral tablets and what have you, got to the point, the PSA was coming down, got under 100, but it got like around 60 and bounced back and forth between 60 and 90. And so the decision was made to refer me to another facility. I went to MD Anderson.
Now, when did your treatment change to Pluvicto, and what did your oncologist say about why your treatment was changing?
Cancer came back in 2015. In 2018, I started at Tulane Cancer Center. So we went different. While I was in the trial, we were using different oral medications and of course monitoring and doing scans and all that, and watching the progress of the cancer. And at the same time, he was running the clinical trials with this medicine. Then the name was called lutetium. And the trials were proving to be very, very successful. This lutetium is the medicine that uses nuclear elements. My study was also pretty much coming to a conclusion, because at the same time what they were doing was monitoring my progress. And he was interested in getting that PSA down much slower. So he invited me and asked me if I would be willing to take the lutetium medicine. Had not been approved yet by FDA, and that's what they were working on, getting that approved. And of course I said, sure. And immediately we started a program. It was like a 8-month program, 6 treatments, and they started me on the nuclear injections. And I go back every 6 weeks and get the injection. And each time the results saw the PSA drop like at 10-point increments until I got down to the final one, and it was finally under 10, down to 6. And it hadn't been there in a long time. And right now it's still hovering around 6. It's not completely gone, so I'm still being monitored. But the new medicine known as Pluvicto is now on the market. It has been approved by FDA. And I certainly am high on it. It improved my situation. I have all the confidence in the world I'm going to live longer. And that was the main objective, is to make sure I continue stay alive.
Wow, what an incredible journey. How does it feel now after all these years since that original diagnosis?
Pretty good, very, very good, because I'm alive and I'm able to do things. I'm a little slower. I'm also 80, but I feel good now. I'm back to doing a few things I can't do as much as I used to, and a lot of that got probably to do with my age, but it's summertime here, so I'm back to doing yard work, and I'm good.
Van, thank you so much for sharing this with me. All the best to you. Well, that's it for episode 2 of SNMMI TV. We'll be back again tomorrow, though, with more exclusive material at this year's annual meeting. But don't forget that you can catch our show on televisions throughout the Convention Center, in your hotel room at the Vancouver Marriott Pinnacle downtown on channel 48 and 40. But you can also catch us online. See you tomorrow.